Hi, this is Trey Lath from OpenHelix. Today's tip of the week is on Phosida, a post-translational modification database. As it says in the second paragraph here, Phosida allows for the retrieval of phosphorylation, acetylation, and N-glycosylation data of any protein of interest. We did this tip about 2.5 years ago and has made some major updates, as you see here in this paper, which is listed in the blog post. There have been new tools and lots more data added to this database since the last tip we did. So let's take a quick overview of the database, and I'm going to do a simple search. Here's the home, where we are now. Here's the search, which we'll get to in a little bit more in depth later, as you can choose your species of interest. There are the tools. There's an added tool with some updates. Here, the predictor for phosphorylation and acetylation, and the motif analysis, which is a new tool here. You can use known motifs or your own motifs, etc. You can do some analysis there. So check these two tools out. They might be of interest and are excellent tools from my quick search. There is a very excellent help section here. You can do it by organism or project related. You can look more about how the prediction is done. So for example, here is some more information and some more data from a reference. You can check that out. And if you need to contact the developers of this tool at Max Planck or otherwise, you can check it here. And they have a nice newsletter. So let's get right to a search. We're going to search in Homo sapiens. Once you choose that, you can choose a protein of interest or by category. We're going to choose protein of interest. In the other tip, we did category. You might want to check that. It's changed a little bit since we did the last tip. But let's do protein of interest. We are going to put in the name, ACTB, and click push return. We have several choices here. We are going to choose the first one here and look at it. So I've chosen a protein of interest. Now once we do that, you'll get more information here for phosphorylation sites. You'll find here some information, for example, component where this protein is found, functions from gene ontology terms, process, and the sequence. You can also go to Reactome, an excellent database, which we have a tutorial on. You might want to check out. You can also, these are the features, you can go to the motifs and domains and find some, check those out. As you see, if you scroll over, find some more information. We can go to other sites. For example, Phospho, Elm, or Swissprot information. And you can find references for this protein here. So you see there's lots listed. Alternatively, you could also look at the evolution of this site and these proteins, the sites in this protein. So for example, here we have protein concentration. No significant alignment is red, one directional best alignment is blue, and two directional best alignment is green. So we see several here. Let's choose PTM conservation. Let's choose K328. And as you see here now, it gives us the site and the species, and it will show you the conservation and the species it's conserved in. Let's scroll down and see some of that. We can do that for any of these sites. You can compare those. So let's go back. So that's a protein search. It gives you lots of information about phosphorylation or acetylation sites, and you might want to check Phosida out if you need to do that kind of analysis. It's a great database and growing and updated. So check it out and come to us next week for our next tip of the week. Thank you.